Okay, in this lecture, you're going to learn about the different mechanisms of disease causation from a particular gene problem. So you might wonder, okay, there is a problem in a particular gene, but how does that actually cause a genetic disease? And it's not very simple, it's actually quite complicated. There are various methods, so many different pathways that can be affected. So we'll learn a little bit about that. There's no easy way of memorizing or learning about the different mechanisms. So bear with me, this is a slightly dry lecture, but just follow along. I don't expect you to remember every single mechanism for every single disease, but just to help you understand how different these mechanisms are and how our body works so differently and each gene works so differently. So let's learn about the molecular, biochemical, and cellular basis of genetic disease. So as you can see in this diagram, there are so many different proteins, so many different pathways that genes are associated with. So in the mitochondria, the peroxisomes, lysosomes, all of these can be affected. Extracellular proteins such as transport proteins, proteases, hemostasis factors, or matrix proteins, hormones, or other proteins in the extracellular area can also be affected. Enzymes, which are primarily present in the cytoplasm, can also be affected, and we'll talk about a few conditions which do affect these. A cytoskeleton can also be affected, protein glycosylation, which is something we'll talk about as well. Cell surface proteins can also be affected, and a number of mechanisms within the nucleus can also be affected should there be a problem in genes. We won't go through each and every single one of these mechanisms, but we'll handpick a few that would be useful for you to understand, just to illustrate the point that there are varied mechanisms of disease causation with genetic problems. One thing it'll be useful for us to all understand is that there are two main different types of proteins within our body. One type is called the housekeeping protein, which is present in virtually every cell, and these proteins have fundamental roles in the maintenance of cell structure and function. And the other types of proteins are the specialty proteins. These are proteins with unique functions contributing to the individuality of the limited types of cells in which they're expressed. When housekeeping proteins are affected, generally the effect is expected to be more widespread. Specialty proteins, the effect can be just depending on what sort of tissues or cells that specialty protein affects, but it can also be more widespread depending on whether that protein itself has secondary effect on other organs as well, such as enzymes, where even though the enzymes actually are produced and might affect some cells or organs, they can also be effect of secondary or downstream effects of those enzymes on various other organs as well. So let's talk a little bit about enzymopathies, or defects in enzymes. Enzymes can be housekeeping, so present certain very generalized enzymes can be present in almost all cells, but there are also some specialty enzymes which are only present in some cells. They're generally considered to be catalysts that mediate the efficient conversion of a substrate to a product. The human genome contains more than 3,700 genes that encode enzymes, and there are hundreds of human diseases that involve enzyme defects. We won't talk about all of these conditions, but we will handpick a few examples under each category to discuss. So in this category, we're talking about hyperphenylalaninemia. Lysosomal storage diseases like Tay-Sachs disease are also enzymopathies, but we won't be talking about in too much detail. Basically, in lysosomal storage disease, what happens is when enzymes are affected, the substrates remain trapped inside the lysosomes, and because these substrates have large size, it prevents their egress, or they cannot be pushed out easily from within the organelle. Therefore, it leads to toxicity within the lysosomes, and these lysosomes can therefore be affected. So let's talk a bit more about hyperphenylalaninemia. So what is the biochemical pathway in these conditions? So phenylalanine gets converted to tyrosine within our body, and it's phenylalanine hydroxylase, or PAH, which helps with this conversion. One of the catalysts is also BH4. So any condition that affects the PAH enzyme can cause hyperphenylalaninemia. conditions that affect the catalyst BH4 can also cause phenylalaninemia. So there are different varieties of hyperphenylalaninemia, but the most common one is classic phenylketonuria. So there is excessive phenylalanines, and then these phenylalanines are then excreted in the urine. So all genetic disorders of phenylalanine metabolism are inherited as autosomal recessive conditions, and they are due to loss of function variants either in the gene encoding PAH, as we talked about in our previous slide, or in genes required for the synthesis or reutilization of the PAH cofactor, tetrahydrobiopterin, or BH4. Or rarely, and there are some other causes, but we won't go in detail about the rare causes. So we'll just stick with the more common ones. So classic PKU is one of the most common conditions in childhood onset, and many countries have newborn screening to detect PKU. So classic PKU, or classic phenylketonuria, results from pathogenic variants in the gene encoding PAH, which converts phenylalanine to tyrosine. Patients with PKU cannot degrade phenylalanine, and it accumulates in body fluids, damaging the developing central nervous system and causing intellectual disability. The neurological damage is largely avoided if we can actually detect it early and reduce dietary intake of phenylalanine. That's why it's so important to detect this early, and that's why many countries have introduced heel prick testing for newborn screening, where it can be detected and we can just modify the diet to prevent all of these difficult effects of phenylketonuria, particularly on a developing brain. Now let's move on to another mechanism of disease causation, which is altered protein function due to abnormal post-translational modification. Post-translation is once translation of the protein occurs. So as you can remember, RNA is eventually translated to protein, and there is some modification that needs to occur after the translation of the protein for the protein to actually be useful. So congenital disorders of glycosylation. Glycosylation is a type of post-translational modification. So if there is disorders of this glycosylation, then that can affect many organs and can cause many genetic conditions as well. Approximately 50% of all proteins and 80% of those in blood are glycosylated and need appropriate sugar moieties added to function properly. So congenital disorders of glycosylation involve more than 160 genes. They're mostly autosomal recessive, although there are some X-linked genes associated as well. Usually present in infancy with multi-system problems, including failure to thrive, liver disease, hypotonia, or interstitial disease. Usually it is protein-losing enteropathy, developmental delay, eye and skeletal anomalies. They can also have immunologic abnormalities and may or may not include brain and or neurodevelopmental abnormalities. Another mechanism is loss of protein function due to impaired binding or metabolism of cofactors. Some proteins acquire biological activity only after they associate with cofactors, like BH4 that we discussed in the case of PAH. Another example is homocystinuria. Homocystinuria occurs due to cystathionine synthase deficiency. Cystathionine synthase is needed to produce cystathionine and then cysteine. The phenotype, or the features, the clinical features of cystathionine deficiency or homocystinuria is dislocation of limbs, intellectual disability, osteoporosis, long bones, so the arms and legs are quite long, and thromboembolism of both veins and arteries. So this phenotype can sometimes be confused with Marfan syndrome, and many people don't get diagnosed easily with homocystinuria. So you can see here the metabolic pathway where cystathionine synthase, if it is not working, then cystathionine is not produced and therefore cysteine is not produced, and there is excessive homocysteine which can be excreted in urine. That's why it's called homocystinuria. Let's move on to another mechanism, which is pathogenic variants of an enzyme inhibitor. So the classic example of this is alpha-1 antitrypsin deficiency, which is an autosomal recessive condition. It is associated with a substantial risk of chronic obstructive lung disease, specifically emphysema, and it can also cause cirrhosis of the liver. In populations of European descent, alpha-1 antitrypsin deficiency affects approximately 1 in 6,700 people, and approximately 4% are carriers. So it is not a rare or too rare a condition. There are lots of carriers, and if 2 carriers partner, then their child has a 25% chance of having alpha-1 antitrypsin deficiency. There are a dozen or so alpha-1 antitrypsin deficiency alleles, but the most common allele that causes the condition is called the Z allele. The Z allele is caused due to the variant P.glu 342 lysine. So glutamic acid gets converted in the position 342 to lysine, and it's relatively common. And when this occurs biallelic, so homozygotic, both the maternal and the paternal alleles both have got the Z allele, then there is a high chance that the person can have alpha-1 antitrypsin deficiency and therefore can end up with emphysema and cirrhosis of the liver. The lung disease associated with the Z allele of the alpha-1 antitrypsin deficiency is due to alteration of the normal balance between elastase and alpha-1 antitrypsin. So elastase is an enzyme that actually degrades elastin in the alveolar walls, and it is necessary to remove excessive elastin so the lungs aren't too stiff. However, if there is unchecked elastase activity, so the alpha-1 antitrypsin usually checks the amount of elastase, it balances it out. But when there is excessive elastase and not enough alpha-1 antitrypsin, then it allows progressive degradation of elastin of alveolar walls. Therefore, the alveolar walls becomes damaged and causes, therefore, emphysema can occur. Infusion of normal alpha-1 antitrypsin is sometimes used in some patients to augment the level of alpha-1 antitrypsin in the plasma in the hope that that can rectify the elastase and alpha-1 antitrypsin imbalance. Another mechanism is dysregulation of a biosynthetic pathway, and an example used here is acute intermittent porphyria. It is an autosomal dominant condition, and it causes intermittent neurologic dysfunction. The primary defect is a deficiency of porphobilinogen deaminase, an enzyme in the biosynthetic pathway of heme, required for the synthesis of both hemoglobin and hepatic cytochrome P450 drug metabolizing enzymes. So you can see here in the clinical expressed AIP, when ALA synthetase is affected, PBG, which is the porphobilinogen deaminase, is reduced. And when this is reduced, the production of heme is reduced, but there is excessive porphobilinogen in the blood, and that can cause a lot of intermittent problems. And this usually is affected when there is certain exposures like drugs or some chemicals or steroids or excessive fasting. So these people, when these insults occur, then they can have all of the clinical features of intermittent neurologic dysfunction. They can suffer from pain, or they can have acute psychotic symptoms as well because of excessive porphobilinogen in their blood. Another mechanism is defect in receptor proteins, and classic example and a common example of this is familial hypercholesterolemia. There are multiple genes, so the gene products, one of the gene products is LDL receptor, which is the most commonly affected gene. We can also have the protein apoprotein B, an ARH adapter protein, or PCSK9. All of these can also be affected. As opposed to the many other genes, you can see the PCSK9, it's actually due to gain of function, which means that there is excessive PCSK9 protease rather than defective or less of the protein. So when there is excessive protein, sometimes the gain of function can also cause problems. So all 4 of the genes associated with familial hypercholesterolemia disrupt the function or abundance either of the LDL receptor at the cell surface or of ApoB, which is a major protein component of LDL and is a ligand for the LDL receptor. So because PCSK9 protease is usually gain of function, there are some drug targets that reduce the enzyme activity, and therefore they can actually help to reduce cholesterol. And this has been used not only for people who have a problem in the gene encoding PCSK9 protease, but also for many other difficult to control patients who have got very high cholesterol and their cholesterols don't reduce easily. PCSK9 inhibitors have been used in these patients in some special conditions to reduce their cholesterol because it reduces the enzyme activity. Now, it's also important to know that early genetic testing leads to early diagnosis and therefore preventative cholesterol lowering therapy and careful diet for these patients. So even in those that we don't know, we can still test based on their family members. If a particular person in the family has been detected with familial hypercholesterolemia, then many other people who may not otherwise be detected can also be tested genetically to find out if they've got the same genetic problem. And therefore they can easily be picked up and preventative therapy can be instituted for them. There are also defects in transport of proteins, classic example being cystic fibrosis, which is the most common autosomal recessive disorder in children of European ancestry. The gene responsible is CFTR, CF transmembrane regulator. The CFTR protein is a regulated chloride channel located in the apical membrane of the epithelial cells affected by the disease. It belongs to the ABC binding cassette family of transport proteins. Cystic fibrosis is due to abnormal fluid and electrolyte transport across epithelial apical membranes. So it affects lungs due to depletion of airway surface fluid. So there isn't enough fluid within the lungs, and that causes problems. They can't actually clear any abnormalities within the lung surface. And similarly, it also affects pancreas, intestine, hepatobiliary tree, and as well as the male genital tract. And this is all due to thickening of the secretions, and therefore the ducts are actually blocked in many of these organs. The loss of CFTR function means that chloride in the duct of the sweat gland cannot be reabsorbed. This leads to increased chloride and sodium concentrations in the sweat. And this can easily be detected by screening sweat test. If there is increased concentration of chloride in this sweat, then that is an indication that there is probably an underlying diagnosis of cystic fibrosis. Another mechanism is disorders of structural proteins. The structural proteins, for instance, in this example that we've used here, is muscle protein. So the dystrophin glycoprotein complex is important for integrity of the muscle and strength of the muscle function. Duchenne muscular dystrophy, as well as Becker muscular dystrophy and some other types of muscular dystrophies, are all disorders of structural proteins. So Duchenne is X-linked, usually boys are affected. The defect is in post-translational modification of the structural protein. And Duchenne occurs earlier. Onset is usually around the age of 3 to 5. There is progressive proximal muscle weakness, so they have difficulty crawling or even standing up and walking. They're usually on a wheelchair by 12 years, and many of them haven't walked at all. Eventually, they might have respiratory muscle weakness as well and unable to breathe without any assistance. And they could also get cardiac complications. Some have intellectual disability too. They produce little to no protein. Becker is a slightly milder variety. It's because of a different allele compared to the Duchenne, even though it is in the same gene. They do express some protein. It's milder than Duchenne. They can remain ambulant beyond teenage years. Another mechanism is pathogenic variants in genes that encode collagen or other components of bone formation. One example that's been used here is osteogenesis imperfecta. So in this condition, people have easy fracturing of bones, even with minimal trauma. It can range from perinatal lethality to only mild increase in fracture frequency, depending on the genes affected. It's generally autosomal dominant, although there are some genes, which I haven't included here, they can be autosomal recessive as well. More than 95% of people have heterozygous, which means only one copy. Heterozygous pathogenic variants in one of two genes, COL1A1 or COL1A2, that encode the chains of type 1 collagen, the major protein in bone. So the phenotypes are influenced by which chain of type 1 procollagen is affected. The disease can be caused either due to diminished collagen production, so milder form, because there's still some normal type of protein being produced, even though the amount is less. So the normal protein is still being produced by one copy of the gene, but it's diminished because the other copy is not working. So that's one type of disease. So even though it is loss of function, it's actually not very severe. Another mechanism of disease is due to structurally defective collagens. So this can be more severe because the structurally defective collagen is actually interfering with the other half, which is the normal protein. So the abnormal protein affects even the normal protein structure. So therefore, even the normal protein, the 50% of normal protein is also being affected. And this is called the dominant negative effect. And usually this is more severe. So the effect of gene duplication and retained function on phenotype. Spinal muscular atrophy is an example here. It is an autosomal recessive condition. It's the most frequent genetic cause of infant mortality, affecting 1 in 10,000 live births, and a carrier rate of 1 in 40 to 50. So it's not too uncommon. The disorder leads to progressive loss of the alpha motor neurons of the ventral spinal cord, as well as the motor nuclei of the lower brainstem. Therefore, the features are hypotonia, muscle weakness, and atrophy of variable severity depending on the underlying genotype. There are 3 main types, SMA 1, 2, and 3. SMA 1 is most common. Symptoms start around 0 to 6 months. They have proximal muscle weakness, respiratory insufficiency, and poor feeding. SMA type 2, approximately 30% of cases. They also have muscle weakness, but starts a bit later, about 16 to 18 months. They may still never be able to walk, but they could still have respiratory insufficiency and dysphagia, which is difficulty swallowing. SMA type 3 occurs in approximately 15% of cases. The onset is anywhere between 18 months to adulthood. They're generally able to walk, but over age they may develop proximal muscle weakness. Respiratory muscles may not be affected in this case. So the most common cause of SMA is a deletion. So as you can see here in the panel B, you can see that the SMN1 gene is absent, which means it is deleted. There is a gene deletion. This is the most common cause, and it can be detected by newborn screening. So there is a new therapy called Zolgensma, which is gene therapy. So just a one-off treatment through intravenous infusion introduces the normal, fully functional SMN1 gene in people who do not have a copy of this gene. And this gene seems to somehow integrate, and it keeps working continuously. So it's a major turning point in the treatment of SMA. We have discussed previously in some other lecture about mitochondrial diseases, so I won't repeat too much about that. You all know that mitochondrial gene is a circular gene. It doesn't, the mitochondrial DNA is a circular DNA. It doesn't have too many genes on it. And pathogenic variants in the mitochondrial DNA and the associated disorders can be inherited. If there is a mother who's got a mitochondrial variant, her children can inherit it. Or it can be acquired for the first time in a particular person as a somatic mutation. So these diseases show some distinctive patterns of inheritance, maternal inheritance. So only a mother can transmit mitochondrial defects to her child. The mitochondrial DNA is only inherited in the zygote from the mother, not from the father, because the cytoplasm, which consists of cytoplasm and all the organelles, are shed from the sperm. So only the egg supports all the mitochondrial function. We've previously talked about homoplasmy and heteroplasmy. Heteroplasmy is when some of the mitochondria within a cell are affected, whereas there are some normal mitochondria as well. Homoplasmy is when all of the mitochondria within that particular cell are affected. When a cell with heteroplasmy then divides, then there is replicative segregation. Some of the cells might get more of the abnormal mitochondria. Some cells might get less or not or no abnormal mitochondria. So there is different segregation. So when it replicates, the segregation is not equal. It doesn't mean that if there is a total of 6 affected mitochondria, it doesn't mean that the daughter cells will have 3 each. It doesn't work like that. It's the division of abnormal and normal mitochondria in the case of heteroplasmy is unequal. That is what is meant by replicative segregation, which means that some tissues will have more abnormal mitochondria and some tissues will have less abnormal mitochondria. And the tissue effects of abnormal mitochondria are also different depending on the utility of the tissue and how the organ is actually functioning. So mitochondrial diseases are quite complex and complicated and may be difficult to diagnose. But just to know that this is also a mechanism of disease causation. So you can see here that the range of affected tissues in mitochondrial disorders and how some people struggle for a long time before they're actually diagnosed with mitochondrial disease. So mitochondrial diseases generally affect tissues that depend on intact oxidative phosphorylation, which means these are high energy kind of organs like the eye or the hearing, so which is the ear. Gastrointestinal tract or peripheral nervous system, endocrine organs, kidneys, heart, or the central nervous system. These are all high energy requiring organs. Included also in here, although it's not been mentioned here, is the skeletal muscles as well.